application under ask a question. Next up is Mrigayu. You're on. Thank you.
All right. Good morning, everyone. My name is Mrigayu Ghosh, and with my mentor, Charles Tracy, and my PI, Dr. Helmut Krämer, we found that the all-nighter mutation decreases count while increasing area of axonal autophagosomes and mitochondria under continuous life. So let's break that down a little bit. We all know how important sleep is in humans, and when we're sleep deprived, functionally we're just not as strong, and we can't complete our daily functions with as much productivity or efficiency. But what we don't understand really is the cellular and molecular reasons for the responses our bodies make in response to these changes in sleep. So one line of research is to use the model systems like the fly, and in our case, Drosophila melanogaster, the fruit fly, to study genes that interfere with sleep. And we can do this because humans and flies both have similar sleep patterns. Normally, they're awake and active for 12 hours of light and then asleep for 12 hours of dark. And in our experiment, this was the control, referred to as LD, for light dark. And we compared the LD to LL, which is, you guessed it, light, or 24 hours of continuous light. And for both of them, we applied the condition of 3 days. So 3 days of continuous low light exposure. LD was compared to LL. The other variable we looked at was comparing the wild type, the wide-eyed fly, to the all-nighter gene. So in flies, this all-nighter gene was identified, and we see that it affects the process of sleep in mutants for that gene. And right now, there's no published data on the all-nighter gene, but the lab has preliminary data showing that the all-nighter protein is involved in the synaptic transmission of the visual system. So our goal was to try and discover the cellular differences in these neurons that could explain these differences in sleep patterns. So as you can see here, here's a schematic of the fly visual system. The R is the retina. The R1 through R6 are the photoreceptor cells, and L. Sorry about that. Yes. So the L right here is the lamina, and L1 and L2 are the lamina dendrites. So the photoreceptor cells, the R1 through R6, their axons actually go through the lamina. So if we look here in this image here, this is actually a cross section from the lamina, and 6 of these photoreceptor cells that are shaded yellow, along with the 2 lamina dendrites, those make up a cartridge. And we can see here that this is a structure of a typical neuron. And so in humans, in humans, our photoreceptors actually don't have axons, and the synapses actually happen on the cell body itself. But in flies, we can see that once the photoreceptor cells receive that light, the synapses are carried down through the lamina. So that's why it's important to study the lamina here. So what I did was I used the software Fiji or ImageJ to quantify these structures. So if we look here, what I did was I quantified the cartridge area, I quantified the mitochondrial area, and the autophagosome area. And in case you don't know, mitochondria are like the powerhouse of the cell. They're what make the energy in the form of ATP, and autophagosomes are these structures that are involved in the process of autophagy, which helps in the degradation of cellular components. And as you can see, using Fiji, I was able to quantify each of these structures, measure them, and then I used Microsoft Excel to process my data and GraphPad Prism to do my statistical analysis. So here I'm comparing wild type LD versus LL, and then here I'm comparing the all-nighter mutant LD versus LL. So now we have results, and we see that in these separated scatter bar graphs here, there's not much going on in the total mitochondrial area. They're pretty much the same when we compare them. But when we go to mitochondrial count, not much difference between LD and LL in the wild type. But there's a significant drop for the all-nighter mutants, suggesting that the all-nighter gene is likely needed to maintain mitochondrial count in response to stress. And when keeping LL constant, we see that the all-nighter mutant has significantly less mitochondria per cartridge than the wild type. And because the total mitochondrial area didn't change, but the count did, we would expect that the individual area has to change. Otherwise, that wouldn't make sense, right? So there's not much difference between LD and LL in the wild type, no significant result here. But there's a significant rise in the all-nighter mutant, and that makes sense based on what we talked about with the area and the count. And then when we keep light constant, so when we keep LL constant, the individual mitochondrial area is significantly higher for the all-nighter mutant, as you can see here, than the wild type. So we did a similar analysis for autophagosomes as well. And once again, not many significant results here for the total area. But when we look at the count in each cartridge, we see that for the wild type, the light has significantly more autophagosomes in each cartridge than LD. And that makes sense. Response to this stress of continuous light, right? If you're having continuous light for 24 hours, 3 days straight, we would expect that. But for the all-nighter, we don't see that significant difference, suggesting that this all-nighter mutant doesn't seem to adapt to this stress by changing autophagosome count. And then when we keep light constant, we see that the all-nighter mutant has significantly less autophagosomes per cartridge than the wild type. And it's a similar pattern we saw with the mitochondria. And similarly, with the individual autophagosome area, we see lots of significant results here. For both the wild type and the all-nighter mutant, light decreases individual autophagosomal area compared to LD. And then for LD or LL constant, either one we keep constant, the autophagosomal area is significantly greater for the all-nighter mutant than for the wild type. So we just discussed these results that you can see here on the left. But the implications of that are, for this third bullet point, the all-nighter gene is likely responsible for responding to stress by increasing autophagosome count. And the fourth bullet point here suggests that the all-nighter gene is responsible for maintaining mitochondrial count and size in response to stress. So in terms of future directions, we could do an analogous comparison of cellular structures in the retina. And here you can see TEM's transmission electron micrographs of the retina. So similarly, I studied TEMs of the lamina. These are TEMs of the retina. We could study the lysosomes and mitochondria here, and we could further understand the transport of autophagosomes to better explain the results that we saw. Finally, I'd like to thank the Krämer lab, my PI, Dr. Krämer, my mentor, Charles, all the other members of the lab, and the National Institutes of Health for funding this project. And I'd like to thank Dr. Goodman, Lynn Tam, and Maria Sandlin from the STARS program, and everyone else in the STARS program, as well as UT Southwestern for providing a world-class facility. And last but definitely not least, I'd like to thank my teachers, family, and friends for their unending support. Thank you for your time. And if you'd like to learn more about the Krämer lab, you can scan this QR code.
Thank you, Mrigayu. That was a great talk. I enjoyed that. Next is